So we're going to enter, based on what you guys know, various techniques of recombinant DNA. So things we can do to manipulate DNA. And the key concept is thermal denaturation. And a close second is chemical denaturation. So these are ways of turning double-stranded DNA into single-stranded. How do you get them apart? How do you unzip it? So that's called denaturation. Then we get into sequencing. This is awesome. It's one of the most incredible techniques you will hear of. Massively parallel sequencing. As most people know today, hopefully know, at the time of the Human Genome Project, it cost several billion dollars, no, $1 billion or more dollars to complete the Human Genome Project in 2003. Today, the entire human genome can be sequenced overnight for a few $100s, around $1,000. Okay, and it's because of methods like these, massively parallel sequencing. And this was all done by small companies, which created really cool ways of getting lots of sequences. Coupled with this was the massive increase in computing power. So all of a sudden, we have laptop computers and workstations which can handle these massive amounts of data, which we'll talk about shortly, create something called cont'ds, or contiguous sequencing. So this is all coming up. So all of this goes into our ability to create transfects, or sequences of DNA, packaged in an appropriate way to enter receptive cells, either bacteria or mammalian cells, and get them to make stuff for us. Okay? That's what all of this is doing. Massively parallel sequencing basically enables genomics. Genomics, the study of gene sequences. Okay. Plasmids and vectors. Vectors. We started off with plasmids. You need all of these pieces in order to make a transfect, so make a recombinant organism, which is expressing genes that are not its own. This is called a transgenic, and that's at the heart of biotech. The restriction enzymes we did, and remember we mentioned CRISPR. CRISPR takes advantage of a system lots of bacteria have to try to get rid of viruses. We have this barrier, which is the nuclear envelope. So in eukaryotic cells, there's a barrier, so that when viruses enter also the outside of the cell, right, there's the life, called the non-life. When they punch through, all of a sudden, if you have DNA in the cytoplasm, this is not a good thing. And eukaryotic cells can recognize this in luncheon. Not so in bacteria. This is its own chromosome sitting there. So that's how this technique works. So CRISPR basically is a method whereby viral sequences that invade the bacteria become learned. So it's a little bit like our immune system. CRISPR members massive, so it will generate a single cut of the genome. Most restriction enzymes will generate one cut in, say, a few thousand base pairs occurring by minute. Vectors, we've talked about plasmids. Plasmids are very, that's this picture right here. Plasmids are naturally occurring. And one of the first plasmids was observed in E. coli. Massive genome, little guy. So plasmids were pieces of extra chromosomal DNA. These are tiny little loops. They're only about 5,000 base pairs long, so they're enough to hold just one or two genes, and that's it. So plasmids auto-replicate, so they replicate themselves. Many species of E. coli can have hundreds of different copies of plasmids, some the same, some different, in their cytoplasm at a given time. Okay, so plasmids, there's lots of them, and these are something that codes for special kinds of sequences. A lot of you guys may have heard of multi-drug resistant bacteria. So we have bacteria today that exist. Many are of the flesh-eating bacteria type. So Staph aureus, for example, which is present on everybody. All of us have Staph aureus on us. But there are Staph aureus that exist. It's totally resistant to all antibiotics known to man. If you get a Staph aureus infection today, it's basically a death sentence unless some antibiotic emerges for So plasmids, all sorts of pieces of DNA, extrachromosomal, small, about 1 to 4,000 base pairs, 1,000 base pairs. Replicate independently. It has something called an ORI, or origin of replication. And you go through all the different elements that you have to have on one of these plasmids in order for it to replicate autonomously, i.e. maintain itself as an extra chromosomal piece and not get lost. Can be used as vectors. So humans basically have taken these, modified them, so that they're better suitcases, better receptacles for us to package foreign DNA and get it into bacterial cells. So we have taken advantage of this and basically altered the sequence. We talked about cutting and inserts. I'm going to go over this more in a couple of slides. I went through this already. Transformation of bacteria, key concept. Transformation of bacteria is massively ineffective. So out of maybe a billion bacteria, you may have 10 or 20 that transform. Ways we have of inserting foreign DNA into bacteria. Remember, viruses have binding sites and sort of molecular injector apparatuses for punching through the membrane, and we don't have any of that. So how do we get a loop of DNA into a bacteria? We have to get past the cell wall, which is pretty porous, and past the membrane, which is not porous. And as those of you taking bio may recall, plasma membranes, biological membranes, highly impermeable to ions and anything big. Glucose doesn't go that fast across it. You need some sort of transfer. Treat bacteria with calcium chloride, plasmid DNA chilled on ice, mix them together, and hopefully some of it will go in. Some of you guys may have done this in high school. This is called competent cell. It's basically a cell with little holes punched in the membrane. Not enough to kill it, but enough to make it pretty sick, and enough to give it holes so that some plasmid will flow through. It's very inefficient. Electrophoresis, Dr. Dorian Osgood is going to go over this more, these types of techniques, in spring, in our spring class. Massive high voltage, remember DNA, strong negative charge. It's acidic, right? It has negative phosphate backbone. So you can rip this thing at high velocities and it rips right through cells. So electrophoresis, massive voltage. You put a plus and a minus pole, hit the voltage. Don't stick your finger in there. I did that once. Pulse massive, and you get some of it punches through. Okay? The whole point is that only a few out of, say, every million or so cells is transformed. So if this happens, picture a field of bacteria, there's millions of these, and there's maybe one bacteria only that has plasma in it, so it has a plus plasma. Okay, now how do we as researchers pick this guy? We want to make this guy grow up and reject everybody else. And remember, this can be about 1,000,000 to 1. The answer is selection. So what we do is on the plasmid, in addition to insulin, and remember insulin was Genentech back in the 70s, sold for $7 billion, I believe, in Roche. Selection markers. So in addition to insulin, all you need is, say, a antibiotic resistance gene. That's the most common, like beta-lactamase. This will hydrolyze penicillin. As a result, you can take this whole ton of bacterial blood, chuck it in a broth that has penicillin. This guy will survive and the others will die. It's called selection. That is how you find the needle in the haystack. I've got some scheme of selection. The other one I'm going to describe is called blue-white. So it's a blue-white transformation. So we'll talk about that next. Very short. So you need some scheme. So on this plasmid, plasmids have a circular map. More on this later. So this is called the plasmid map. It's a circle. Remember, this is somewhere around 4 to 5 kb. On it, and we're going to have features on this map, and I'm going to draw all those out. Usually there's a base pair number 1, and it goes all the way up to 4,000 or 5,000, however it is. And you can put inserts in here at certain sizes. Everything got it. You need selection. Transform cells on different antibiotics. Does not select the plasmid contain foreign DNA. Research plasmid. More on that in 1 second. So put that on the blue-white selection. I mentioned, I think this, I can't remember what class, it was either this class or not. We talked about LAC-Z. This is the LAC operon in bacteria. Remember, the way bacteria operate is we have transcription factors. The eukaryotic systems have transcription factors. Transcription factors absolutely essential to get transcription. Remember, again, every cell has the whole full complement of 22,000 genes. But each cell is only expressing about 5,000 or 6,000 at a given time, which is roughly a quarter. Usually this is driven by transcription factors. Bacterial systems don't have this. You've got very few, if any, transcription factors. They have operons. So what they're doing is responding to what's available in the media outside them. And as soon as lactose or some unusual sugar, milk sugar is not that common in the living world, relatively rare. So as soon as that appears, the bacteria want to start expressing genes that will enable them to bring lactose in. Remember, lactose, big sugar, it's going to be across the membrane very, very slowly, if at all. This is biological membrane permeability. Very slow, if at all. So it needs a transport system to get lactose in, okay? So one of the genes is LAC-Z, and LAC-Z codes for beta-galactosidase. And that's the heart of this whole thing. We're now in blue-white selection, and there's a couple slides like this. So LAC-Z, this is a typical name for a gene. So this refers to the gene on the bacterial chromosome. Codes for beta-galactosidase. Here's the ACE at the end, which means it's a hydrolysis reaction. Milk sugar is glucose and galactose hooked together. Galactose is also C6 sugar, but epimerized is slightly different. So beta-gal will recognize that. Okay, so we're taking advantage of LAC-Z for our selection. In other words, we're going to use this gene to help us find the needle in the haystack, the 1 or 2 bacteria that actually took up the plasmid of interest. I mentioned antibiotic resistance. Blue-white is the second method. Okay, very, very common. So what do we have? DNA cloned in a restriction site in the LAC-Z gene. I'll show you a picture. I hope you made it clear. When it is interrupted by inserted gene, LAC-Z gene cannot produce functional beta-gal. when X-gal, X-galactose is an artificial lactose. Remember, lactose is a disaccharide. I can't draw structures, you guys don't know this. But lactose sugar is a disaccharide. And I'm just going to show it as a sketch. Blue meaning glucose and galactose. Right? Remember, all those polymers, including fat oligos, all are hydrolyzed to split them and condensed to bring them together. Condensation builds polymers, water is removed. So hydrolysis is the reverse. So you add water across this, H2O, and you need beta-gal to catalyze this. Beta-gal, this is converted to a glue. These can now be metabolized right through central metabolism called fermentation. Okay. So lactose disaccharide, what this means is you can trick this thing you can trick beta by instead of having glue, we're going to put in something that is a colored dye. And this is going to be carefully engineered so that beta-gal cannot distinguish this from lactose. Now when it does a hydrolysis reaction, when the pigment no longer is in this glycosidic bond form, it emits light, absorbs light in a particular wavelength. In this case, it changes color and it becomes blue. Turns blue. This dye is blue. Blue color. This is the heart of blue light selection. Remember we got it? Another slide, hopefully. Non-functional laxine will be white because this gene is disrupted and it cannot hydrolyze that bond. Okay. Go slowly through this, no panic. And here's bunches of plasmids. These have all been transformed. This picture looks like it's totally worthless, but these colored dots on there are blue. Here's plasmid. Remember I showed you the map. All plasmids need an ORI, origin of replication. This is showing double-stranded DNA. This is about 3,000 base pairs. So you must have an ORI. If there's no ORI, the plasmid cannot replicate itself and will be lost. ORI is basically a spot where DNA pole can bind. DNA polymerase binds here, and then zip this and make copies of it so it replicates. With no ORI, it is lost. Okay? Then there's something called the multiple cloning site. More on that later, don't we? Multiple cloning site. And this multiple cloning site is stuck right in the middle of LAC-Z gene. Trying to assess consciousness. Okay? Now, if you don't cut this, and you don't stick human insulin in there, there's an upstream region, there's a promoter and an operator here. RNA pole will bind. Beta-gal is made from the LAC-Z gene, and the colony turns blue when you chuck in this altered lactose. Everybody got it? Okay, now we color one. We cut this, remember it's showing an overhang here, so this is FLR1 or some restriction in it. We cut this. We put here generates cohesive ends. This is been cut. We put some human DNA, shown in black. The human DNA has been engineered so that the cohesive ends and overlap are identical. This enables hydrogen bonding. So this now pairs in that slot. Got it? You chuck in something called DNA ligase. Ligase connects this so that there's no double-strand breaks, and this now is a sealed piece. Everybody understands this to this point. Okay? Plasmid, cut it with restriction enzyme. Make sure that the human piece has identical overlap. Usually this is done by cutting with the same endonuclease. Just so that these are the same. Chuck in ligase and you seal it up. You now have the original plasmid engineered to have this insert of human DNA in it. Bear in mind that the restriction site where we're cutting must be in frame, right? It has to be, remember, there are 3 reading frames. 2 give you gobbledygook, and 1 gives you beta-gal. You want to be sure that your protein is in frame so that the amino acids are all stacked up correctly and you don't end up with a terminator in here. Got it? So now our RNA pol binds and it rips off this gene, but with your protein in it. The blue part is no longer functional. Beta-gal has this huge thing stuck in the middle, and it's not functional. You often put terminators in here, so the back half isn't even transcribed. No beta-gal. This colony, any bacteria that has this plasmid, will now be white. It will not turn blue, because beta-gal is dysfunctional. Everybody's still alive. Ligase seals this whole thing up. Lactose non-functional. Somehow, electrophoration, calcium chloride, freezing and heating, this guy gets in. Remember, this is 1 in a million down here. You grow this in culture. You grow this thing in culture. You select this guy by looking at all these colonies. You now have all these colonies. These are all blue now. The kinds of blue bacteria. This is after you put in color gal. Remember, this is glycosidic bond. And the beta-gal thinks it's lactose and it hydrolyzes this. Turns blue. All of a sudden, I don't know how to draw this because I can't draw white. Pretend this is white. There's one guy here. This is white column. I can't draw in white or you wouldn't read it, be invisible. This guy has this gene, beta-gal, disrupted because there's an insert. As a result, it is white. It is no longer blue. You pick this guy, going with your little loop, you pick this guy out, and you grow him up, and you make billions and billions of plasmids. Do you understand? This is selection. This is a key method for selection. It's called blue-white selection. We actually will do this in lab in the spring. Okay, study this slide. Be sure you get it. So assume you have a plasmid with lactose gene containing a restriction enzyme site, just like I showed, somewhere in the middle of it. Plasmid also has antibiotic resistance. Following transformation, you grow the cells on agar plate containing antibiotic. And here are the results. Which colonies have the inserted gene and why? So in this description, they're doing a double selection. This is typical, because trying to find 1 in a million is hard. So first, screen it for beta-lactamase or antibiotic resistance, say to penicillin or ampicillin or anything you want, and then grow them on a plate. Which colonies do you pick? 1, 3, and 6. These are white. These colonies, that beta-gal or the lactose gene has been disrupted and is no longer functional. All right.
Why does it turn blue with the presence of lactose in its functioning?
Because lactose looks like this. Okay. Remember, the bacteria is not normally eating lactose sugar. It looks like this. What we do is we have chemically made Professor Bosey will go over this when you guys take more go. We made chemically this. Oh, shoot, yeah. Reverse the direction, but let me just, I want it to be the same as the picture. I'll try that. We make synthetically dye. Beta-gal can't tell them apart. So as soon as beta-gal is expressed, it hydrolyzes both of these. We feed the plate this. You put this dye substrate right on the plate. Colonies that express beta-gal cleave this and it turns blue. So the cleavage is not blue, it's white. Once you hydrolyze this, it turns blue. So in other words, we engineer an artificial substrate which resembles lactose so that when it is hydrolyzed, it turns blue. That is why. So you have to put the substrate in order to get the blue color. And obviously, any colony that cannot turn blue is not making beta-gal, and it cannot hydrolyze this type of silicon. Okay, all of this is exactly what here's Cohen and Boyer did. This is Genentech, part of Roche. Boyer and Stanley Cohen completed cloning expression of human insulin in 1978 for the treatment of diabetes. Huge breakthrough. It's revolutionized and basically introduced the entire industry. Within a few years, Biogen, a lot of other companies, biotech companies, surged, and today there are literally thousands, many of them right here in the Providence area, and they will, some will live and some will die. Okay, Genentech purchased by Roche for $47 billion. I was off by $40 billion when I said that. $47 billion. These guys became instant billionaires at that time. Today, hundreds of recombinant proteins are on the market for treatment of all kinds of disease indications, etc. First human protein expressed was insulin. The second was growth hormone. Growth hormone, for example, is used to treat dwarfism. For people that are producing defunct growth hormone that have a defective gene, remember SNP. A SNP mutation in growth hormone, so it's not functional. You can give and administer growth hormone only in the youth during the growing period, and they will be normal stage. Clone human insulin DNA in the plasma, bacterial cells that are used to synthesize protein products to clone the gene. You can generate lots of pure protein by this method, and a lot of this is we talk about in the spring semester biomanufacturing. So you grow up some recombinant culture, you scale it up into massive fermenters, you make tons of stuff, and you sell it on the open market, you make billions of dollars. Generate lots of pure protein. Growth hormone, prior to recombinant technology, was isolated from human cadavers. Insulin, you know where insulin came from before Boyer and Co? Cattle. Well, even though the sequence is very close, it's not exact, and there's a lot of other horrible stuff in cattle's serum, for example, in pancreas. There's viruses, bovine diarrhea, all kinds of horrible stuff. So people are being treated with insulin are often being treated with something much more horrible. So generated from human cadavers, obviously there's a danger of viral, pathogenic contamination, etc. Because whatever killed this person, it may not have been on the road, it may have been some infectious disease that it may have gotten a viral infection. Practical features of DNA cloning. Okay, so these are all the types of features that you would find on a typical plasmid. Let me go through this slowly. I have a slide coming up with a map on it. It's PBR 320. One of the first plasmids that's ever appeared on the market. You can buy all the stuff. You can buy all this stuff. And which we all have. And bring it into your lab and start chopping it up and designing constructs. So all vectors, whether it's a plasmid, there's many, many different types of suitcases. Some look like foot lockers, they're huge. Duffel bags, and others are very small. So plasmids is pretty small. Viruses get much bigger. Yeast artificial chromosomes called YACs, big. You can put huge stuff in there. Plasmids, small pieces. You can only put like insulin or a single little image. Small enough to be separated from chromosomal DNA of host plasmids. Easy to do, 3 to 4 to 5,000 base pairs is good. ORI. Must have ORI, site of DNA replication. No ORI, this thing will be lost. Must have an ORI, it must be functional. Copy number refers to the number of plasmids in the cell. Normally this is small, but you can manipulate things. We'll talk about that. I don't think we'll talk about it in this class. We'll hear about it more to the one. There are ways of making your plasmid be, instead of 3 or 4 copies in a bacterial cell, hundreds. Hundreds of copies means huge protein production. Multiple cloning site, MCS. Recognition for several restriction enzymes in which insert is cloned. These are generally in frame. So that the start AUG, these things, even though they're span, are in frame. Selectable marker gene. You must find the needle in the haystack. You've got to find one out of millions. Remember, massive voltage and punching holes, very inferior. RNA pol promoter sequence. So you must have some sort of system so that your gene will be expressed in bacteria. DNA sequencing primers. Mostly for sequencing, which we'll talk about. That's coming up when we talk about PCR or rich DNA. Any questions? This is on the next slide. This is POC. POC comes from the University of California. All plasmids have a small p, which is the code way to do it. So p means it's going to be small bacteria in plasmid. They all look like this. If you look at it, this is called replication here instead of ORI. Here's LAC-Z gene, MCS, multiple cloning site. What it means is we've engineered LAC-Z so that EchoR1, BAM8, SMA, a whole bunch of endonuclease sites are present in the beginning when this gene is being transcribed. That makes sure they're all in frame so you can cut, stick your piece in to disrupt LAC-Z, and your gene now gets expressed. PBR, we did this when I was in college, when I was in grad school. So PBR 3.2 was available and POC appeared about the same time. First widely available plasmid ancestor of this University of California. Circular piece of autonomously replicating DNA. Means it's capable of making copies of itself in the chromosome of bacteria. Originally evolved by bacteria, I think I mentioned this. Where did plasmids come from? E. coli, again, express these. E. coli is the one we know of. Many other species of bacteria have plasmids too. E. coli express these in order to express toxic genes that killed off other E. coli. As a result, it allows you to exploit the food source. If there's a piece of rotting meat and you want to consume it, right, and it's off the side of the road somewhere, say roadkill, you want to be sure that everybody else trying to eat it dies. More food for yourself. That's how these things evolved. So E. coli appeared, which had, they're called eosins. They're basically bactericidal genes. Well, we've exploited them by getting all the bacterial sided stuff out of there and sticking in all the things we want. That's how these things fit in. Got it? So this is a perfect small-sized suitcase. It replicates itself. You can put any gene you want in. You can select it. Here, block. Beta-lactamase. Penicillin is this polycyclic structure. and beta-lactamase inactivates it. So this thing kills, hydrolyzes, and inactivates penicillin. Any bacteria that has taken this plasmid up will survive in the presence of the penicillin. Needle in the haystack, right? You can pick these guys. And then pick somebody who's turned from blue to white and no longer make the dye, turn the dye blue. Certain abuse of autonomy originally evolved in bacteria may express antibiotic resistance genes or modify to express proteins of interest. Let's pause here. We'll take a 8-minute break. Reconvene at 10 o'clock. I wrote this on almost all of your quizzes. Semi-conservative DNA synthesis. Remember, this was the only question I'm going to review for you. Remember, this was Watson and Crick, and Crick actually proposes this in the Nature paper, the seminal Nature paper. Double-stranded DNA, I'm just going to write it like this for simplicity. Remember, because of the hydrogen bond pairing between A's, T's, G's, and C's in the middle, this can be opened up, maybe in the middle, like in a bacteria. Opened up, this exposes all these little hydrogen bonds. Now it's been unzipped. This can be further separated so that the whole thing is open. Right to the end. Then you make new strands. And Crick points out that wherever there's an A, a T is going to be the complementary one. Wherever there's a T, an A, a C, a G, and G, a C, right? So the mononucleotides can all fit in here and will be weakly hydrogen bonded to all the hydrogen bond pairs, right? Remember, there's a leading and lagging strand, but that's a subtlety. Then when this stuff gets zipped all together, you're going to end up with, which I wrote on almost everyone's quiz. A couple people got it, but most people didn't. Replicated instead of one genome, you now have two, so the cell can divide. It has one old and one new strand, as long as you wrote something down about one old and one new. I gave you credit. Okay. So this is called semi-conservative, because each new strand has an old and a new. And as we'll talk about, through methylation, this is part of epigenome, remember, which is methylation acetylation patterns, not sequence. The cell can tell the difference between old and new. You can tell them apart based on methylations. That way, which is coming up later, if there's a mismatch, if there's a mistake, the cell will know that this is correct and this is wrong. Just tell the difference between old and new. Some of you may have been wondering that. So that's how mismatch repair works. Methylations, you can tell which is the old strand. New strands are never methylated, they come out brand new. That little subtlety coming up later. Everything covered. Semi-conservative. This is proposed by Crick in the Nature paper. Now I have to, at 10:30, I'm going to stop and we're going to switch over to aseptic technique. This will be with some of you that were attended biomanufacturing this spring. And also crime scenes. We have two really cool labs that are starting today that take a couple periods. So I have to stop and give you basically the pre-lab during lectures. A solid hour with okay, vectors. These are different suitcases. The plasmids I described is one of many. And there are tons of these. And companies have come out with new ones and whole new designs with different features every year or two. So there's lots and lots of different types of little suitcases, packages, which enable you to get your gene in, the gene of interest. We'll talk more about how you get that in, and then get the whole thing into a bacteria. Plasmids we discussed, small, 6 to 12 kb is typical. Bacteriophage or phage vectors. These are massive. This is like lambdaphage. Lambdas are well-known E. coli virus. So we take the virus, we're going to chop out most of the viral sequences and stick in our own. So this is called a vector. bacteriophage vector, 35 kb. Remember average gene size? Usually somewhere around 4,000 base pairs. Generates a protein of about 1,000, 1,200, something like that. So this is massive. This is several genes or many genes. Backs and yaks. These are artificial chromosomes. This technology came out relatively recently. You can go up to hundreds of kilobases and up to mega base pairs. It's getting massive. This is like entire bacterial genome. This is in yeast. Remember, yeast is so cool because it is a eukaryotic cell. So it has a nucleus, and it makes things similar to our cells. Yeast is a very close relative. Fungi is right next to animals in the tree of life. And then TI vectors is a particular type used in plants, like agrobacterial cells, which we'll talk about when we get to the plant section. I'm going through them one at a time. Bacteriophage vectors clone up to 25 kb, lambda genome is linear. Bacteriophage is getting bigger. Remember now, I already went over plasmids, so I'm not going to go over that. But this is bacteriophage, much bigger, 25 kilobases. This is huge. This is maybe 7 or 8 genes. Cloned DNA is inserted into restriction sites where common chromosomes packaged into viral particles. These infect E. coli. What you usually do is you smear a plate with E. coli, put it in the oven and warm it up so that it's a confluent log. So the entire petri dish, agar, is now full of bacteria. So you roll up a plate, and you have here a complete log of E. coli. Every available centimeter of this plate, this is called the log of E. coli, or some other bacteria. Every center of the plate is covered with E. coli. And it has kind of a slimy white appearance and it smells. E. coli is coliform, so it's found in human stool, for example. Fecal material, it's not pleasant. Well, anyway, grow E. coli up, and then you infect it. I have a picture of this coming up, so this picture will become relevant. Cosmids. These came out again about 25, 30 years ago. COS are cohesive ends of lambda DNA, plasmid orient replication, gene for antibiotic resistance, DNA is cloned in the restriction site. Cosmid is packed with viral particles which will affect E. coli, bacterial colonies grow on a plate, screened with antibiotics. The fragments can be big, 20 or 40 kb. So plasmids are getting much bigger. Remember, instead of 6, 7, 8, 9, and 10, it's about double or triple that. Some plasmids feature shared by either plasmid or bacteriophage. So hence the name cosmid, the cohesive ends of plasmid. This is a E. coli lawn, and it smells. And onto this lawn, you cover it with recombinant phage, bacteriophage. Bacteriophage, or phage, is a virus that infects and kills bacteria. Okay. You have your lawn of E. coli, and then you squirt on top of it a solution of your virus that you transport. This, instead of a little small suitcase, great big suitcase. It's got much bigger genome in it. Every little hole, see hole there? A hole is where the virus has infected the E. coli and killed it. So it's lysed. As a result, you have a little hole here. It clears it. That means that your recombinant bacteriophage infected an E. coli cell right in the middle. It killed that cell. As you guys may know from viruses, as the E. coli dies, it bursts out, and out now come hundreds of viruses. This keeps going. You end up with billions of viruses in that zone, cleared zone. You take a little tiny pipette, it's called like a P1 or P2, which are only pipettes about 1 or 2 microliters, and you suck up a little bit of fluid from there which contains recombinant virus. All bacteria dead. That's called a plaque. It's called a plaque here. And if you suck that up and then look at it, it looks like this. So these are billions and billions of phage. Every one of these will have encapsulated in the head viral genome, bacteriophage genome, with your insert in it. Another option to use in a plasmid, plasmids or so. Bacteria expression vector vial, high level protein expression, bacteria RNA polymerase, binds to the promoter, transcribes the insert, makes the protein. Insert is something you want. This is usually a human gene for medical purposes. A biotherapeutic protein is purified using all kinds of biochemical techniques. Column chromatography, this is all called downstream. So downstream purification allows you to take a huge vat full of bacteria, mash them all up, and purify insulin out and get rid of all the bacteria garbage in the real world. That's called purification. Problems, bacteria expression. This is why we have so many. Because a bacterial plasmid is massively restricted. There's not too much you can do with it. You can do some stuff like make insulin, but not much of it. Sometimes bacterial ribosomes cannot translate eukaryotic protein. The protein is not folded correctly, since bacteria does not have organelles for processing. Remember, the whole train, I think I went over this in my class. At any rate, the whole train of protein production in the eukaryotic cell is all compartmentalized. So remember, a lot of ribosomes, if you guys remember the video for bio, a lot of ribosomes are studded on rough endoplasmic reticulum, which is clustered all around the nucleus. So as soon as mature mRNA, remember, it has to be processed, exons spliced, introns removed, 3' poly A, 5' cap. It has to have all those three things. An exit, usually, often it goes right over to the ER, which is sitting right over the nuclear envelope, arranged in stacks. Protein is made in the lumen, it moves to smooth ER, Golgi apparatus, and it is being processed the whole way, glycosylated, trimmed, cut, altered, often post-translational modification, where acetates and phosphates and all kinds of things are added. Bacteria doesn't do any of this. It has none of these compartments. So that means that if you're trying to make a fairly sophisticated human protein, it will likely not be made correctly. Okay, this is a prokaryotic system. Enter yeast. Yeast will do it correctly. So it's eukaryotic and has all the organelles. It's not possible to use the system with eukaryotic genomic DNA. Why cannot you use eukaryotic genomic DNA in bacteria? For example, in a small plasmid.
Too complicated. Is it too complicated?
Correct, but why? What specific complexity is present, is inherent in a eukaryotic gene that is not present in bacteria? Virtually every eukaryotic gene has this failing.
Certain things not being used.
How are eukaryotic genes constructed? What is a unique feature of a eukaryotic gene which is not present in bacteria? I'm thinking more of the genome sequence itself. How are genes organized in eukaryotic DNA? Yes, more specifically. Come on, genes. It's not a, because remember, I can take, even though chromosomes are huge and long, each gene is a tiny little piece, and I can cut that out, and I can put it into a plasmid, so it is circularized. What else is different? How are most eukaryotic genes constructed? There's something unusual about them. I just went over this. Eukaryotic genes, eukaryotic makes a pre-mRNA. It is massive, long. What is the first thing that happens to it before It can exit the nucleus. I just said it. There's three. That's one.
Left in the tail and the cap.
That's it. So here is a typical bacterial gene. Remember, about 4,000 base pairs. Typical bacterial gene looks like this. This is 4 kb. Okay, 4,000. Typical eukaryotic gene looks like this. It's supposed to be double stranded. One gene, 4 kb. Little piece here, little piece here, another piece here, piece here, piece here, another piece here, bigger piece here, etc. There can be 30, 40 exons. You can't cut the whole thing and stick it in a tiny little plasmid. Right? This can be hundreds of kb long. Got it? Key problem. Most genes look like this. Remember, we thought there were hundreds of thousands of genes in humans. There's only 22 because of exon splice variants, shuffling. Okay? You cannot do this. This thing can be hundreds of kb long. You need a trick. So genomics is not possible. Human genes are usually split into exons, and introns can cover thousands of base pairs. Difficult to find insulin coding sequence in genomic DNA. If it's split up, how can you, if you know this whole sequence, how do you know which of these pieces are coding for genes? How do you know? How do you know that there's a 20,000 long base pair intron there? Right, in bacteria, it's dead easy. You just find the AUG, right? You get your -10 and -30 regions, cat losses. You find the AUG and you just run down in here until you hit a terminator and you're done. Eukaryotes, enormously challenging. Okay, got it? It's a fundamental difference between eukaryotes and prokaryotes. Complex gene structure. They're fragmented, often fragmented. It's very hard to find. Backs. Massive, low copy number plasmids. This is like not even a foot locker, it's like a car. Huge amount of space. Contains genes code F factor, controls bacterial replication. Huge pieces of DNA. This is up to hundreds of thousands of base pairs. A lot of this was used during the Human Genome Project because we had 3 billion to go, and if you're sequencing little 4,000 base pair pieces, obviously you're never going to finish. But with this, at least you can start getting something approaching a megabase, so a million base pairs. Yaks. Beauty of yaks, eukaryote. Eukaryotes can splice. And eukaryotes do have a nucleus, and they do process proteins and transcripts. Miniature version eukaryote chromosomes, ori or rep. It's often called rep, meaning replication. Ori is origin. 2 telomeres. Telomeres are sequences at the end. More on that later. Selectable markers. Centromere. This is the part in the middle that allows replication of yaks, segregation of daughter cells. Best for cloning massive pieces up to several megabases. That's million base pairs. Well, you can span the human genome with what you need a lot of them. Still, megabases. Human Genome Project, small plasmids from E. coli introduced into E cells, Saccharomyces cerevisiae, which is brewer's yeast. This is the model organism for single cell fungi. TI. This is a soil bacteria, normally occurring plasmids found in soil bacterium. Bacteria infects plants. tDNA from TI plasmid inserts in a host chromosome, so it's like a retrovirus that actually integrates into the host. tDNA codes for auxin, weakens the plant cell wall, forms what's called a tumor or a crown gall. So if you look at an old oak tree, it will have this great big knobby growth on the side of it, and that is all based on a agarobacterium that has infected it and produced this crown gall. Scientists use TI vectors to deliver genes into plants. Plants very hard. Plants are, well, they're plants. They're eukaryotes. They have tough cell wall. Okay, DNA libraries, this is really the key. So when I've been talking, I can go on. You guys will remember talking about it's like the red pen is to be remembered. So we had a bacterial plasmid, and here to here was the LAC-C gene. LAC-C member codes for beta-gal. Then we cut this, so it had a staggered end. And then we insert a piece of host DNA with a base pair overhang that matches, right? Remember all this? We insert that, and we end up with this. This piece is overlaps with that, so we end up with this. We ligate it up and seal it, and this is some, say, human gene. Human gene. Say insulin. Got it? Remember this? Question. How do we get this? Where does it come from? Knowing the sequence alone from the Human Genome Project is very helpful, so we know the exact sequence of A, T's, G's, and C's. But how do we actually physically get this piece? Now you might say DNA synthesizer. DNA synthesizers are good, they're not that good, although they're pretty very good today. There's another simple way to get this.
Enzymes to cut it out.
Pardon?
Enzymes to cut it out.
Correct, but remember again, lots of human genes are in pieces. So the probability that you're going to have a restriction enzyme that is exactly cutting out all these exons is totally unrealistic. So how do you get the gene out? How do you get this piece? The answer is library. John?
Okay. No, I was going to say CRISPR because it's more specific in one, but
True. CRISPR will help you, but still, even good idea. Still, if you cut at both ends of one of these massive genes filled with introns, you still have a gigantic piece.
Okay.
And how do you get the coding sequence out? That's called a library. So in order to do this, scientists and now companies, library, have constructed DNA libraries. DNA libraries are fragments of DNA or complementary cDNA, more on that in just a second, which contains human genes. And they're usually present in bacteria. You can order them from companies and they're cheap. So you can say, I want, out of 22,000 genes, I want gene number 17,526. And they will send you a little tube with recombinant bacteria that often is it'll be a virus, often a virus they send you. And you can take that infecting coli, suck up the little plaque, and you now have a virus that has your gene in it. That's how you get it. Okay, and more on the pieces, the axons, in just a second. Two types of libraries, genomic and cDNA. The most powerful and the most useful to us by far is the cDNA library. Basis of next motion slide. Hope that we get to it. I was hoping to show you some cool technology, but I don't think it will. Okay, I think if you focus on the details, you're probably going to end up using. So, how do you make a cDNA library? Chop up human DNA, get these pieces inserted into some sort of plasmid, and produce bacteria which have all these different pieces in it. As I just went over, companies will sell you bacteria for each of the 22,000 genes. There's a problem with splicing, and we'll talk about that later. Okay, easier route. Harvest mRNA from cells. If you get, for example, an embryonic stem cell, almost all the RNAs, all the genes are being transcribed in an early cell. So harvest mRNAs, make sure these are mature mRNAs. All the splicing has been done for you. Right? Cell's already done it. Remember, if it gets out of the nucleus, all introns are moved. It's all been spliced together. 3' poly A, 5' cap. Has to be there. So take mRNA. There is an enzyme, which this is one good thing human immunodeficiency virus did for us. HIV is a retrovirus. It is an RNA genome. First step that happens, God forbid you guys are infected with HIV, but if you are, is RNA, which is the genome of this virus, enters your cell, is now in the cytoplasm. Not that unusual for RNA to be in the cytoplasm, right? We have lots of RNA in there that's all being translated. Reverse transcriptase converts the RNA into DNA. It's called RT. mRNA is digested away. You fill the copy out. You now have a double-stranded piece of DNA which codes for the mRNA. This will have poly A tail, the complementary strand has T's, and then we'll have some sort of 5' cap. In this case, we're going to put a link on, I'll talk about that. The beauty of a cDNA library, no introns. What you see is the coding sequence. And it has a bunch of T's, A's, and T's at one end. Remember, poly A, 3' A. So the complementary 5' A will have T's. Everybody got it? Have linkers, chop this up. Now create a library of 22,000 cDNAs. No introns. All this, the huge picture I had with black lines and little blue boxes is now all blue. So all the blue boxes are hooked together. So these are all axons. Right, this is a mature mRNA. So the 3' end will have a whole bunch of things. And the 5' complementary will have things. This is the 3' poly A. And this is the 5' cap. You can actually engineer some stuff up here so that it's useful to us. Got it? The beauty of the cDNA library, no introns. It's only coding sequence because the cell has done all the work for us. It's chopped out all the garbage, which makes it impossible to do something. Instead of cloning a piece this big, you get to clone a piece this big, which has only the coding sequence in it. Remember, the average gene is a few thousand base pairs. And some of this is looking pretty good. You can stick this in plasma, you can stick it in some cosmos, things that are loaded. Genomic libraries, chromosomal DNA is chopped up, many fragments, include the entire genome. Vectors digested the same enzyme, so that the overhangs are complementary. DNA ligase, ligate all this up. The common vectors are used to transfer bacteria. And theoretically, each bacteria will contain recombinant plasmid. You collect all these bacteria, you separate them all, you figure out what pieces they have in them, and you sell this to people. So any piece of the human genome you can buy. It's called a library. It's just like a library of books. Disadvantage of genomic libraries. Introns are cloned. Majority of the genomic DNA is introns and eukaryotes, so majority of library contains non-coding inserts, introns in the middle. Many organisms have very large genomes, so searching for gene of interest is difficult, time-consuming, and generally a massive waste of time. Nobody does it because of this cDNA library. mRNA already spliced. Now you guys can say, but Dr. Chris, depending upon what tissue it is, maybe you're only going to get one splice variant. You won't get to see all the different ones. That's true. But you can look at different tissues and look at different mRNAs and see all the different types of sequences made from a given gene. Right? mRNA will vary often between cell types. So a voltage-gated calcium channel in the heart muscle is going to be different from immune-responsive cells. Reverse transcriptase catalyzes complementary single-stranded DNA from mRNA. This is called complementary because it's an exact copy of the mRNA. mRNA is degraded, DNA fulfills the whole thing in. You now have double-stranded cDNA. No introns, small, only going to be a few thousand base pairs. this thing up here, non-cDNA library, is massive. Short linker, double-stranded contains rich enzyme site, cut with some enzyme, cut vectors with the same, ligate them up, transform them, you're ready. I'll try to get some total amount. See how I'll stop short. cDNA libraries, a collection of actively expressed genes in cells or tissues which mRNAs isolate, introns not there, or spliced out. Creating discrete isolate genes, disadvantage is difficult to make cDNA library, the source tissue abundant amount of RNA from gene is not available. Okay, this should be cell. If you take any eukaryotic cell, say it's muscles. So here you've got some cardiac. This is a cardiomyocyte. It's heart muscle. And you look at all the mRNA that it's expressing, and you purify the mRNA out and say, I'm going to make now a cDNA library. Problem? What do muscle cells mostly make? Anybody remember one of the muscle proteins?
Is it actin and myosin?
Actin and myosin. So 95% of the mRNAs you get are going to be actin and myosin. They're going to be muscle proteins involved in contraction. It's not that useful. You want to get maybe all kinds of other genes, not actin and myosin. But you can pick other cell types. You usually pick a cell type that you think is going to be expressing the gene of interest, so that it's a significant proportion of the mRNA that you purify from that cell. Got it? A nerve cell, similar. It's going to be making lots of sodium pumps and voltage-gated channels, etc., because it's all electricity. It's all a battery. Managed collection of vectors, introns not going to be created, disadvantage. Difficult to make, RNA is not available. Assume the gene involved in increased muscle mass, expressed muscle cells increase growth hormone. Best source of DNA for seeking a library of muscle cells express growth. That's just saying select the cell or the tissue for the gene that you want to get. Okay, I know this is brutal. Do your best and I'm going to stop after this. Key concept. This whole chapter, unfortunately, is full of keep forgetting those. Okay. So work with you. Here's a huge piece of double-stranded DNA. Double-stranded DNA. This is huge. Huge one. Here's a small piece. And this one we're going to say is about 20 centimeters. Okay. How do we get these apart? So how do you denature or convert double-stranded DNA into two single strands? Variety of methods. It's called denaturation. The main method, heat. If you heat this up, these guys start to wiggle. Remember, heat gives you more kinetic energy. The balls are bouncing around, atoms and molecules are bouncing around faster. It starts to breathe. So as you heat this up, it's going to breathe. It tends to breathe where there are lots of A's and T's. A's and T's only have 2 hydrogen bonds. G's and C's have 3. So G's and C's are more stable. So regions rich in AT open up first. So these will start to breathe. So as you go up to about, let's say, 40 degrees centigrade, this guy completely floats apart. It's totally denatured. This will have regions, and little bubbles are forming, where lots of A's and T's are present. Also, if there's mismatches, so something's screwed up in your sequencing, and there's a mismatch, that area will tend to open up faster too. As you keep increasing this, and you go up to 60, 70, 80, up to about 90 to 95 degrees C, all DNA completely denatures from millions of base pairs down to little pieces. They're all separate. This, you guys, is the basis of almost all DNA sequencing. So all probing, like chips, Affymetrix chips, where you examine sequences of people to decide whether or not they should take particular drugs, or susceptibility to certain types of genes, like the BRC, the breast cancer gene. You do it all using thermal, also chemical. You can add in NaOH base. So if you add in Y, or NaOH, sodium hydroxide, the negative charges tend to blow DNA apart, and this is nature's too. So you can do it at room temp with a variety of agents. Base, also denaturants, such as solvents. Solvents will begin to dissolve, solubilize the hydrophobic base pairs in the middle, and they'll come apart. So all of these methods, chemical or heat, can be used to turn double-stranded DNA into single-stranded. And that's what this picture's showing. So as you cook this thing up, this is some piece, it's maybe a 30 or 40 mer, and you're looking at how much of it is open. You start off with none of it because it's all double-stranded. As you near about 53 degrees, the precise sequence, it's called the melting temperature, will depend upon how big and long this is and the kinds of stuff that it's dissolved in. If you have formalin, some kind of solvent, it'll reduce the temperature. If there's lots of A's and T's in here, it'll reduce it. If there's a mismatch, melting temp will reduce. Got it? So different environmental features will dictate at what point 50% of this is single-stranded. That's called the melting temperature. For this particular piece, it's 53.5. This is a good sized piece, maybe a 40 mer or so. 40 base pairs together. You keep going up another few degrees, 100% denatured. Totally single-stranded piece. Okay, let's just do this one and stop, and I'm going to switch gears. 10 base pairs. This is slightly warm room temperature air. These melt. So 10 base pairs comes apart at room temp. 30 base pairs, about 60 degrees. 100, about 70. 10,000, 75. Human genome, somewhere around 90. Keep heating it up. They'll all come apart somewhere south of boiling temperature. This is critical because that allows you to do all kinds of things such as PCR, polymerase chain reaction, which is the focal point of today's lab. Yeah, today's lab. You guys we're going to set up a PCR machine. I'll talk about that. Okay? Thermal denaturation. Key to all these technologies. We're stopping on this slide, and I'm now switching to aseptic technique. As I said, you guys that already took this in the fall are going to be somewhat bored. But I'm sorry, it's not that What the hell? And this stuff's all uploaded on Sakai. I did it last Friday. We're not going to get to see any of this. Oh, we'll get to see PCR. Aseptic technique. Wow. Today we're doing 2 labs. I'll explain more on this later. Roughly a third of you are going to be doing aseptic technique. I'll explain about all that when we get to that. Okay. Aseptic technique is the method whereby you guys in a lab can prepare and grow mammalian cells, for example. Mammalian cells are eukaryotic cells. They grow tortuously slowly. So typical replication time for a mammalian cell like a CHO, which is a Chinese hamster ovary cell, or any mammalian or human cell, is about 24 hours. So one cell will go into 2 every day. Bacterial cell can be 20 minutes. So within 1 day, you will have millions of bacteria and maybe 4 or 5 human cells or CHOs. Got it? To minimize this, you have to work aseptically. That's why we have the sterile transfer hoods, which I'm going to go over during lab, when we meet the laboratory. I have to start this now, otherwise the lab won't finish until 6. So, how do we keep the area clean so that there's no bacteria and no viruses there and it's clean? And the answer is to use a aseptic transfer hood, which you guys, when we did, remember when we made media? We made media last week, and the area was clean for you with isopropanol. And Doreen, I think, had everything set up, and you guys went in with your little filtration flasks, and you did media manipulation. You sucked out 10 mils, added in serum, etc. Remember all that?
Yes.
Okay, that's what the focus of today's lecture is. So how do you keep it clean? Definitions: aseptic versus sterile, levels of cleanliness, controlling contamination, sanitization practices, aseptic practices, gowning. I'll briefly talk about SOPs, which are standard operating procedures. These are things written out in books or now online that you follow in order to do certain things. Gowning, flow of personnel materials, monitoring environment consequences. That's the next 40 or so slides. This goes fairly quickly. Understand purpose of aseptic techniques, how to apply them. Again, this is repetition for some of you that were here last week. Handling of sterile objects so as to maintain sterility. This is what you were doing with your filtration apparatuses last week. Clean room conduct prevents further contamination of all its more precise behavioral guidelines. Microorganisms exist in much of the environment. As you guys know, every one of us humans has about 50 trillion cells that are us, and we have about 400 or 500 trillion cells that are not us. This is bacteria, mostly in your digestive tract, on your skin. So every one of these, you do this in micro labs. So you take you can take a sterile Q-tip, go over your skin, plate it out, and you grow up Staph aureus, yeast, everything. Okay? The whole world of microorganisms is living on every one of you. Hair, our skin cells that are lost, bad cough, you're spewing out all sorts of infectious stuff. Have the potential for prolific growth. They can beat out a eukaryotic cell every day of the week. Introduced into the process during manufacturing, microorganisms will cause any of the following: utilization of nutrients. So in other words, they steal glucose from the cells you're trying to grow. Alteration of growth conditions, inhibition of desired culture, production of toxic substances, production of substance reaction or degrades desired product, such as proteases that are secreted by bacteria. To control microbial growth and prevent this occurrence, sterile and clean environments, it's imperative that personnel strictly adhere to the principles of aseptic technique. You guys will be doing this today. Sterile, devoid of all life. We believe planet Mars at present appears to be sterile. Could be wrong, it could be some exotic type of organism. All equipment, media, water, air. Inadequate sterilization of anything used in the culture process invites contamination. Growth media in its container must be sterilized as soon as media is prepared. You guys did this with the filtration apparatus. Vessels covered as much as possible. Cover should not be placed on other surfaces. Once you put something down and pick it up, that thing is now contaminated, even if you scrub this with isopropanol. Somebody will have coughed or perspired or sat on it or something, and it's now not clean. Avoid excessive movement or talking. Do not mouth pipe that. This is a total no-no in all labs around the world. Avoid contact with sterile equipment, surfaces, instruments. All personnel working with the near-grown culture should be appropriately gowned, which is why we have you guys gowned and wearing gloves. Especially critical too. Exposing the culture to environment, do so in an area with little turbulence and as free from microorganisms as possible. Clean room contact and aseptic technique. You've got a bunch of you will be working in the cell culture room, which is adjacent to the main lab. So we'll send over, I think, 1, 2, 3, 4 people will go more on that later. Protect product from contamination that might be harmful to patients. Humans. Biggest problem is each one of us. Every person that goes to that hood and you've got your gown on and your gloves on, you are the biggest danger. Remember, all of us are covered with microorganisms, hair, coughing, etc. Contamination can be controlled through proper behavior and use of aseptic technique. Humans are the worst source. We set the specific practices and procedures, and you guys can follow this if you want. It's all, I uploaded it all on the The goal of minimizing contamination by pathogens or, in fact, organisms of any kind. Asepsis, absence of pathogenic organisms in a clinical setting. This is why those of you that have worked in a hospital or have been in hospitals, people are walking around, staff members, with isopropyl alcohol, exactly what you guys made last week in the solutions lab. Remember, you made 70%. They had 70%, and they wipe everything down, and it makes it aseptic. So most everything is killed in 70% isopropyl. Clean is removing dirt. This is using, a scrub brush. When you go to Amgen, you could literally eat off the floor of the manufacturing facility. It is squeaky clean. That's because it is cleaned constantly, and it is sterilized, usually with bleach. Sanitize, reduce the number of microorganisms to safe levels. Nothing is completely clean. The instant a human enters there, whether they're coughing or not, the area is no longer clean. It's no longer aseptic. Disinfect, remove most microorganisms not highly resistant. These processes, things like spores, can survive long processes. These processes are usually not sufficient to prevent infection. How do we limit cross-contamination? Counting. This is showing what looks like a red blood cell, and this is showing I have no idea why. No idea what that is. Flow of materials and equipment, sanitization of equipment, autoclave, for example. Gowning at Amgen, when we go to Amgen, everybody will have little boots. You wear little boots so that your feet are covered, and you have men with beards. You have to wear a beard cover, and ladies have to do something so you pile your hair up. We'll all have on hairnets, beard covers, unless you don't have a beard, fully gowned and gloved when you go into Amgen. Okay, because you are the biggest threat to a multi-million-dollar operation. Gowning. Traveling in the manufacturing area looks just like this. It's all part of gowning. We go to this in the spring. Clean rooms. Purpose, prevent contamination. Achieve purity, safety, and efficacy for the product that we're trying to make. Okay, this is called a surveillance plate. So what you do, and Amgen does this routinely, I think it may even be a daily basis. They take agar filled with nutrients so everything can grow. They open it up and they set it out in the production area for a period of time. And it's all an SOP, standard operating procedure. So people do this, it's set out for maybe 20 minutes, then they put the cover on, they go back, let it grow up and see what's there. If it looks like this is bad. This is bad. There's mold, there's fungi, it's covered, it's crawling with stuff. This should be an example of an unclean area. Okay? It may have one or two microorganisms that will be allowed, but it cannot have the whole plate covered. We're doing really well. FDA. It's called 21 CFR Part 211. Control of microbial contamination. Appropriate written procedures. These are called SOPs, Standard Operating Procedures. Designed to prevent objectionable microorganisms in drug products, not required to be sterile, shall be established and followed. Bacteria, staph, viruses or flus, spores, fungi, yeast foot, yeast, Saccharomyces, Mammalia, etc., all examples. Particles, all coming generally off of people. Hair, covered with bacteria. Skin flakes, You guys know that every day we slow off like 1 million dead skin cells. And that's why it's good to launder your sheets every week. Break on it. Launder your sheets every week because all the dead cells that you lose when you're asleep at night all fill up the bed, and then they fill with mites. So little mites eat them. So as a result, mite population goes up. Mites are microscopic, very, very tiny. You can see them. Many people are allergic to mites. There's mite feces and mite carcasses and stuff. It's important to get rid of all these types of things. Skin flakes, covered with bacteria, clothing, soil, oils. All these things are sources of contamination. Equipment, supplies, people. Personnel with no activity emits up to 10,000 contaminated particles per minute. It's just you sitting there, just like you are now. Personnel running emit up to 10 million particles per minute. There's a lot more air coming in and out. Sneezing produces 1 to 200,000 aerosol droplets filled with pathogens. Right? We do this in micro. You thaw your mouth out, you plate it out, there's lactobacillus and a bunch of stuff that's harmless, and there's pathogens in there, every one of us. Okay? So sneezing, all this type of activity you have to limit when you're at the hood. Let's stop there. 10-minute break, reconvene at 11 o'clock, and then we'll finish this up. Areas contaminated, especially production. We use aseptic technique, which is not in its sterile. I mean, it's as good as we can get. We cannot thoroughly sterilize a manufactured protein drug product. Problem. Protein is susceptible to nature and cold or get ruined, along with your attempts to get it clean. So in other words, you cannot sterilize the product. You have to make sure the environment in which the protein is made is as aseptic as possible. Okay? So protect the environment, protect the product from people, and then rely on it being safe. You can test it with especially medicines. This is showing another plate covered with horrible stuff. 5 levels of cleanliness. Class 1 is the cleanest. This is like Amgen production floor. Class 5, gown area, suite outbox, stored glassware. Suite outbox, so things that move into a production facility usually go through a portal, so it goes through a protected area. So it's the dirty outside, clean inside. Everybody on the inside is going to be fully gowned, and it'll be cleaned with bleach every day. So 5 is dirty area, and 1 is the clean area. Class 3, suite in-lock, cell culture suite recovery. Contamination, big issue, bacteria, yeast, mold. Mycoplasma, this is a type of very, very small bacteria. Very common contamination. Talk about more in the spring. Cost company money, tons of man hours, and obviously patience. Well, what is this? This is looking at dissolved oxygen is on this axis. Dissolved oxygen is usually around 19%. Why, you ask? Because air is about 90% O2. So the air that you pump into the media, usually foaming it in there. You have enormous air jets in order to keep everybody growing and happy. It goes along, all of a sudden you see a sudden crash. It means probably something else is growing in there besides CHO cell or whatever mammalian cell. Declining nutrient levels, low pH, cells are dying, all evidence of contamination. It's called a spinner class. You'll see it in the spring. Spins, media in here, to inoculate it with mammalian cells like CHOs or DEX. We're going to use DEX in the spring. This is a hybridoma. This has kind of a granular look. typical of mammalian cells. This is good. This is bad. This is plant bacteria. Tons of stuff growing in there. It'll smell. Redundant equipment, backup cell lines. Worst case scenario, this is classic in industry, talk about this. Standard operating procedure. Batch records. Second person verification. This is all a hallmark of production. Environmental processing, designed to monitor manufacturing errors, determine load of microorganisms, rather than just rely on cleaning everything with bleach and isopropanol. A classic example is you go with sterile Q-tips and gown personnel, and they go in there and swipe an area and put it on a plate and say, how much stuff is actually here? How much stuff is great? So determine the load of viable organisms in a given area. It will never be zero. Might not be zero, but hopefully it will be within limits. Total particles and air and bioburden on equipment and facility surfaces is kept minimal. Data gathered provides means to determine whether manufacturing area is in a state of control. You suddenly see a huge uptick, and you're seeing tons and tons of microbes. Something bad has happened. You know, there's a dead mouse in an air duct, or something horrible has happened. Okay? Or there's a live rodent living somewhere on the floor. Essential for preventing microbial contamination of the product. Water, soil, dust, air, people. People, the greatest source of contamination because they shed large numbers of particles, coughing every time you speak, talk, or breathe, urinating particles, as well as skin. Follow procedures which govern gowning and flows or airflows. So all production facilities have slightly higher pressure. The pressure is kept higher, so when you open the door, everything flows out, right? Because you don't want outside contaminated air going in. So it all has, it's called negative pressure drop as you move out of the containment area. Unusual circumstances, leaks, floor damage, like cracked tiles, anything that's not smooth or organisms can collect. Clean, organized workstations, clean up still as soon as possible. Okay, that is the end of the aseptic technique. And a portion of the class will be doing that today. Next is this. You guys have this, it's uploaded. This is crime scene. So this year, I don't think we have an actual crime scene. In the past, we usually have a TA. I think Tara was picked. And the TA lies down on the floor. There's a big fake blood stain. And this represents crime scene. Here, crime scene. So we have a crime scene which is fictitious, and there's a victim. And I think in this case it's Tara. So Tara's tragically was stabbed, and there's blood, and we're going to try to figure out who the perpetrator is. And there are 4 suspects: myself, Dr. D, Ed Bosey, and I think the 4th person is Joel Chandler, who's the chairman of our department. So we have 4 suspects who may possibly have done this dastardly deed. So you guys in the lab today, it's a 2-part lab. So you're going to set up your PCR tubes, we run PCR overnight, and then you run gels next week. You will be able to determine who is the perpetrator. Okay, so now we're talking about forensics. So kit from BioRad. called a gel. We'll talk more about that later. And it's designed to show that it's fingerprints, so you can identify who did what. Real world connections. This is crime scene, which you guys are all going to do this week and next. Showing all different kinds of disciplines. Crime scene, PCR basics, kit tend to be introduction to PCR, more on PCR later. Standards, rule of profiling PCR, about 2 45-minute sessions. Set up PCR reactions and what you do today. DNA profile. So DNA profiling is the heart of crime scene. So you find, as I said, like a rape victim or a body off the side of the freeway, dumped in the ditch, and it's, I don't know, too often a raped woman. But you may find particles of blood on her clothing, and if this blood is not hers, you can now have, as soon as you line up suspects, Dr. Chris, Dr. Doreen, Dr. Bosey, and Dr. Chandley are the suspect. You can say, who matches this? Okay, called DNA fingerprinting or profile sequencing. So evidence collected at crime scene, blood spot, cases of rape, semen, vaginal semen, and then show all kinds of tissue. It's the beauty of something like bones is you can get genomic DNA sequence from Neanderthals. So remains that are thousands of years old. The DNA is very stable. Blood spot on clothing, you extract out genomic DNA. You now have the DNA sequence, hopefully, of the perpetrator. We'll talk about this more later, probably in the spring. So, sample obtained from crime scene or paternity investigation. So in this case, it would be saliva, a cheek swipe, or blood sample from infant, which the mother says this is the this man here is the father. The father says, the guy says I didn't do it. Well, you can prove that he is or is not the father. It's called paternity. Okay. Extract DNA, quantitate it, amplify STRs, short terminal repeats. Work with me a bit, and I'll explain this later. Separate detection PCR products, look at these alleles, genomic determination, comparison of sample to other samples. If match occurs, comparison DNA profile with population databases, and you say, in some cases, there are no perpetrators. The case I'll go over was Colin Pitchfork in the UK, where 2 girls were raped and killed, about 12 or 13 years old. Semen samples, there were no suspects. So a whole village that was nearby, all the males were profiled and the perpetrator was identified. Okay, so you can identify just about anybody from DNA. Crimes investigators search areas of genome unique or anonymous STR, short terminal repeats. This is the heart of forensics. There are many, many loci. Loci is location on the genome which has a particular feature. So it turns out a lot of us have repeats. This is 4 base pair repeats, TCAT, TCAT. This is a SNP. So some of you will have 1, 2, 3, 4, 5, 6 repeats. Others will have 5, 6, 8, 9, 10, 11, 12, a known number. But the whole human population will vary as to the number of repeats they have at a specific position. This is called TH01. This is one of about 13 positions scattered throughout the human genome. Everybody got it? This example has 6. This is variable. The population is not the same. More than 20 known TH01 alleles. So that means there's maybe 4 or 5 all the way up to 20 or 25 or 6. Each individual inherits 1 allele from each parent. So you may get 6 from your father and 14 from your mother. So as a result, if I look at TH01, I will see in every one, remember we have paired chromosomes, 1 paternal, 1 maternally inherited. I will see 6 from one sex parent and 13, say, from the other. And it should be random. So you guys will all have different sequences at the TH01 locus. Everybody got it? Mrs. Smith has 1, 2, 3, 4, 5, 6. and the father has some big number. I'm not going to count this. Okay, we use PCR, and I'll talk more about PCR later. PCR is an amplification technique. The PCR allows you to take a given sequence of DNA, and by PCR, this is called polymerase chain reaction. PCR. We'll turn this into millions. In a few hours, brilliant method. Okay? 1 in the millions. So all of a sudden, you can do standard run-in gel electrophoresis, which some of you may have heard of, and you will now see a span or spot. Whereas here you don't see anything, there isn't enough. Okay? PCR means amplification of a given sequence. PCR, polymerase chain reaction, makes copies of a specific piece. It's based upon primer sequences, which we select. And remember, whole human genome is known. And we know most of the variants. So every one of us has about 3 million different sequences. Those are called SNPs. And that's the 0.1% that differentiates every human on this planet. 3 million out of 3 billion, about 0.1%. PCR allows you to look at one specific piece of data by making copies only of that piece. It's like looking for a needle in a haystack and then making a haystack out of the needle. So making millions and millions of DNA. This is just showing DNA profiling. Which suspects cannot be excluded. All DNA data is always worded like this. You do not identify perpetrators. You prove that somebody could not have done it. Now, there are cases where perpetrators were found out of a huge group, for example, in the Colin Pitchfork murder in the UK, a huge group of males, and the individual was identified, the person later confessed, and other data went into making this guy known to be guilty. Okay? But generally, you exclude people, but you can find that one perpetrator out of millions that are not known. Suspects are included in investigation. DNA profile matches the genotype spot at crime scene. Suspects can be excluded if they do not match. So if you see at TH01 locus, you see blood from the victim. Remember, it'll have 2 loci from your mother and father, so it'll have 2 different alleles. If it's 4 and 16, and I take some suspect, let's say it's me, and I don't have 4 or 16, then obviously I'm not the person that did this. So I can be excluded. Well, this is what you guys will be doing in lab, and I'm going to go over this more when we get to the pre-lab lecture in exactly 1 hour and 12 minutes. You take bunches of tubes, crime scene DNA. This is blood spot from poor Tara Gizak, who was brutally stabbed to death. So we take crime scene. It's coming from her lab coat or something. A, B, C, D is Dr. Chris, Dr. Osgood, Bosey, and Dr. Chandler. Master mix, more on master mix in just a second. Amplify it, then you run out bands. So in this particular case, if we look at crime scene, this is blood found on the body of poor Tara Gizak, who we all know and love fondly. She has this pattern. This is not her blood. Normally, you always have to have victim's blood. So you take a sample of their blood and make sure this is not hers. So if she was stabbed, you're going to see a huge amount of Tara's blood there. She may also have skin under her fingernails from scratching during the fight before she finally succumbs to the lethal blow. Sorry to be so graphic, but it is a crime scene. And you can take the skin out from under her fingernails and get genomic DNA, PCR, and identify all of the STR, short-term repeats. Got it? So in this particular case, whoever is number C is the likely perpetrator. Note that A, B, and D can be excluded. C cannot. The C is looking guilty. If other evidence supports the C being the perpetrator, then you get to nail this one. Or woman. I'll go over all this later. I'm not going to do this now. PCR2, so I can go over this later. This is all details. Okay, PCR. How does PCR work? Let me explain. I have time. You can ask time. This is okay. So, huge piece of DNA. This is some genomic DNA that came from blood spot on dead parent defects lab coat. Okay? This is called the template. Every PCR reaction, you must have a template. You need a Neanderthal bone. You need, some restaurants, this is cool, comes up later. They say, we're giving you special harpoon salmon from Nova Scotia, and you're paying, a huge price in order to eat this in a restaurant. You say, I don't think this is Nova Scotian salmon. You can prove it. Take a sample of the fish, and if it does not match the database, then these guys are, screwed. Template, number one. You must have a template. Oh, same thing with illegal hunting. So a guy has a ton of meat in his freezer, and you say, I think that's bear meat. In some states, it's illegal to hunt bear. The guy says, no, it's not bear. You say, yes, it is. I can prove it. Okay? Here's a tiny little piece. We're going to call primer. We need two of these. Three. Primer one, primer two. Everybody got it? I understand PCR. These concentrations are approaching millimolar. You have trillions of these for every one of those. Right? Trillions of the little guys. You and I make these. You can order them from a lab that synthesizes little pieces of DNA. These are small. They're usually about 20 birds. These are little. These are only about 20 long. This, being human genome, or terrorism, is 3 times 10 to the 9th base. Your entire genome. Coming off of that spot of blood on your coat. Or my blood, if I was to check that. Put this into a PCR machine. This is going to go into put that in there. You add something called master mix. Master mix has primers. It has all 4 DNTPs. What's in all 4 DNTPs? Come on, steady.
Are they used by the PCR reaction to build the actual chains of nucleotides? Like they're various.
What is DNTP? If I wrote down what is DTTP, would you all know what that is?
Thymine.
Thymine triphosphate. This just means nucleoside triphosphate, and there are 4 of them. DTTP, DCTP, GTP, and DATP, right? This just means they're all 4 there, so I don't have to write it out every time. N just means nucleoside. All 4 must be there, because this spot of blood or skin cells under her fingernails, she's beating off her attacker, brutally attacking. We'll need all 4 of these to get the sequence correct. You need a bunch of other stuff. The second critical thing you need is something called TAC DNA polymerase. Must have all these or the reaction will not work. This is asked multiple times on quizzes as well as exams. This is critical that you understand this. All of this must be in here or will not work. Template, 2 primers, 4 DNTPs, TAC DNA, and other stuff. Buffer compost, magnesium, you need a bunch of other stuff. pH control. Okay. So you take this, which you guys are going to do in lab. Okay. All this goes into this is called master mix. And to master mix, you add A or B or C or D. These are samples of the four suspects, Dr. Chris, Dr. Dorian, and Joel. Okay? You're going to have four tubes, one for A, B, C, and D. Presumably represents our DNA. PCR. I've got to erase stuff. Can you guys write all this down before we get to this? I've got to erase this stuff. Okay? PCR relies on thermal denaturation. Heat the sample up, and this will come apart. Okay? So usually, you heat this thing up. So step one is we're going to heat it up to about 90 to 95 degrees C. What happens at these high temperatures? Who breaks apart? Which? This one, all of them? Everybody. This is so hot, this is near boiling. All DNA comes apart. These are all now single stranded. Tack DNA Tack Q DNA pole. This was isolated. This is a DNA pole, DNA polymerase, isolated from Thermus aquaticus. It's coming from hot springs. This organism survives up to 95 degrees centigrade, near boiling, and it was found in geysers, I think, Yellowstone National Monument. Geysers, the water was, people went, oh my God, this is sterile. Who can live in here? This guy. So organisms have adapted so that they can survive at 95 and thrive at about 70 degrees centigrade. You know, this is like 180, and will kill any human within a few seconds. This organism can survive. Its DNA is stable up to 95 degrees. The DNA does not die. Because of this, Harry Mullis, which I'll show you in a few slides, got the Nobel Prize for conceiving of the concept of PCR. Nobel Prize was talking somewhere around the Then cool it down, and you cool it down to some key temperature. It depends. So number 2 is cool to usually at somewhere around, say, 45 degrees C. This is highly variable, and it's going to depend upon the sequence of the primers and the temperature at which they melt. Because there's trillions of these, and only one of those, when it comes together, the probability is, let's say, blue is complementary to here. Well, this now is melted apart. Pretend this is one line. Melted. Blue goes here. I'm going to put a little arrow on it. 500 feet. And green will go here. 20-mer, absolutely unique. Remember, 20-mer will define, just like CRISPR, this will appear uniquely in the entire genome. It's trillions to one. Okay? These will come first because there's many, many of those for every one of these. You then warm it up to about 75, 80 degrees, specific temperatures will come in. TAC makes a copy of this. So TAC, you guys remember, all DNA poles, they need a little primer piece for eukaryotic DNA replication. It's RNA, remember, made by RNA primase. Little short piece. You need the little piece because there must be a free 3' hydroxyl to add the next DNTP on. Must be a free end, or you can do squat. So the DNA pole can bind on this, but unless there's a little piece, it won't start. Once the little piece is there, it has all 4 DNTPs, have plenty of energy and time. It goes and it puts out, it cuts, makes it a complementary piece. On the other strand, the other guy is making this, right? So at the end of the day, from this long one and this long one, you end up with this piece. Here's the original primer. And this piece. You do a cycle. You warm it up to 95 again. You cool it. You do this over and over again. Every cycle will increase this sequence by 2. Do you understand? First cycle gives you 2. Second, 4, 8, 16, 32, 64, 128. And after 20 cycles, millions. That's what Carrie Mullis realized. He was backpacking in California with his girlfriend. They had just reported on TAC. And he suddenly goes, oh my God, TAC works at the exact temperature where DNA denatures and reanneals. That means by using this DNA from this organism, which is super stable and does not die at high heat, I can do this. I can turn one copy into millions. Oh, I have to show you. Here's the video. I think we're actually going to make it. Nothing. Anyway, where are we? We just talked about PCR. You guys should be on the animation slide, and I apologize, I have no idea what's wrong. Okay, so this is showing the temperatures. PCR reaction, 94, 52, 72. So you guys in lab, we're going to program 94, 94 degrees, 52 degrees,
Yes.
70 something? 72, correct?
Mhm.
Okay, let's review real quick. 94, single-stranded, everybody. Genomic templates, little primers, everybody single-stranded. There's too much heat there. So this denatures everything. 52, usually selected as the TM or the melting point for the primer, the little guy. The little guy is somewhere around 52. So this is usually around the TM. This is the point at which 50% of the primers have annealed. Usually the TM of primer. And usually pick your primers. There are 2 of them, remember? 2. So that they're close. You then warm it up to 72. Why 72? Because Thermus aquaticus, which is where TAC DNA pole came from, is most active at around 70, 75 degrees centigrade. This grows in hot springs. This guy makes TAC DNA pole is maximally active. Envisioned by Carrie Mullis backpacking with both. You now go from 1 to 2. Cycle it again. Melt. 52, 72. Now you have 4, 8, 16, 32. Within 20 cycles or so, you have millions. And I apologize, I can't get this thing. This is the next slide showing it denaturing. Primers annealing. Primers anneal at 52. You guys should see green and blue. And the little green ball is TAC, DNA pole, recognizes the 3' end. Remember, with no primer, with no little primer on the DNA piece, TAC pole cannot bind. No polymerase will work. It needs something to start with. Hence RNA primase in eukaryotic cells. Everybody got it? Cycle 1, 2, 3, you end up with from one little blood stain and maybe 1 or 2 copies of a whole template, you end up with millions of copies right where the 2 primers are selected. So this will go, after many cycles, to millions of this piece right here to here. And every piece have a little blue primer at one end, and a little green primer at the other. Okay, you'll have millions of those. These are designed so that you're spanning one of the 13 loci of short terminal repeats. This is all FBI, so the Federal Bureau of Investigation has established what these 13 are, and it's called cont'd, which is the identification system for humans. That shows the gel, add a bunch of stuff, shows you pipetting. Electrophoresis I can go over next time because I can't get this thing to work. We're on paternal lineage testing, bunches of stuff, M&Ms, showing the wheels. Shoot, I don't know how I'm going to help you guys with this. Okay. Everybody have this down. 94, 52, 72. On quizzes, say somewhere around 90s, somewhere around 50, and somewhere around 70 to 75. There are 3 tests. You cycle. This is done using a thermal plate, so we have a heated plate. And, you can control the heat to within seconds. So usually let it go for about a minute, and each one of these steps is about a minute. The whole cycle is about maybe 3 or 4 or 5 minutes, and you repeat it. So in a few hours, you can get millions of pounds. Have you done that? Now, let's say we're looking at this TH01 loci. This is in your handout thing, so you have to be able to answer this question. Let's say we're looking at one locus. There are 13 STRs in the federal FBI database called the cont'd database. Every locus, you have 2, maternal, paternal. And they will likely differ unless your parents are siblings, which is illegal in this country. So likely this will be different. So for 13 loci, you have a total of 26 different patterns. Mother and father, so you're going to have 2 variants in each position. That's what the M&Ms and the 1/6 is showing. Okay, flip down, real world pop. Flip down to cont'd. Can you guys flip down? It's flip down to slide number 47. It should say cont'd, which stands for Combined DNA Index System. And it shows you, I'm sorry, this doesn't work. It shows you all 23 chromosomes from 1 to 22 plus X and Y. And you'll see that there is a different loci of these short terminal repeats in every single chromosome. Almost every one has one, about half of it. Notice that there's AMEL in both X and Y. This is good because male sperm will have either an X or a Y in it. So that's good. This allows you to identify those. With 13 STRs times 2, because every one of you has 1 maternal and 1 paternal, may be same, likely will be different. So with this system, 13 loci generate a high power of discrimination. The next is showing you a microchip, and a gel, and then that's it. These 13 loci, with 26 variants that each one of you had, is sufficiently to uniquely identify each human on Earth. So your loci will be exactly identical, to nobody, so it'll be different. Similar to a siblings, but not close. Sibs will have a closer match, because you have sibs such as their brother and sisters. You have both same mother and father. But still, you will get a different assembly of the 23 from your mother and 23 from your father will be random. It's different. Identical twins, exact same. That's the only exception. If it's identical twins, you can say it is one of these 2. Okay? Everybody got it? I apologize. That's what you need. It worked. See you guys at 12:15 right here. I'll give a brief pre-lab, explain how we're going to run lab today, and then we'll head down to lab and do crime scene. Good luck.